Okay, this is part 2 of the protein synthesis lecture. We just talked about RNA, and now we're going to move on to transcription. Remember that protein synthesis is the process of making proteins. Why is that important? Because all day and all night, your cells are making the proteins they need to stay functioning, to stay alive. Proteins do all the work inside the cells. They build, they repair, they digest, they help to keep invaders out. So your DNA's job is to make the correct proteins based on signals that it gets. And we're going to talk about how. The 2 steps involved in protein synthesis are first transcription and then translation. Transcription, to transcribe in general, not in biology, to transcribe means to write down, right? If I want to see a transcript of, you can see a transcript of a show, and they'll send you, all the words that were said in that TV show. Okay, and that's just a written down version of what someone said. So, in this case, transcription involves reading a gene on a DNA molecule, making a copy of sorts of that gene, and that copy can then leave the nucleus. So, the purpose of transcription is to solve a cellular dilemma. DNA, remember the analogy we started to make? DNA is like a teacher. We mentioned this in part 1. If you didn't watch part 1, go back and watch it. Stuck inside of the classroom. Legally can't leave, right? DNA is locked inside of the vault of the nucleus in eukaryotic cells, which protects it, but that produces a problem. The machines to build proteins are out in the cytoplasm, but the information, the blueprint to build the proteins is in here. So transcription solves the problem of getting information from the nucleus out. And that information is in a copy called mRNA, this pink thing here. Messenger RNA. Notice it's single-stranded. And so here's how it works. All right, I'm gonna send you a link to this animation, which is going to help you a lot, I think. But here's how it works. We're going to open up the gene that we want to make into a protein during the first step of transcription. An enzyme, it's shaped like this blue guitar pick thing, this faded blue guitar pick here, called RNA polymerase, is going to read the DNA letters. Read A, T, C, G, A, G, A. And when it reads the letter, it's going to make a complementary strand. Okay, so when RNA polymerase reads an A, it grabs onto an RNA U, which pairs with DNA's A, and it adds it onto the strand. Then it reads the next letter. It reads a T, it adds an A. Reads a C, it adds a G. Reads a G, it adds a C, etc., etc., etc. And then this red thing here is called mRNA, messenger RNA. It's capable of now leaving the nucleus, okay, and going out into the cytoplasm, where we'll do translation. Okay, so RNA polymerase makes mRNA, which is a messenger molecule that can be used for the next step. Of course, there's more to it than this in transcription. I'll give you a little bit more information. Now, you're not making every gene. Let me go back for a second. Here's your DNA, a real simple version of it. You have, let's call it 23,000 genes in there. Not all the genes are turned on at once. You don't have to use them all at once. Like if you have a recipe book, you're not making every recipe in there at once. You might have a dessert and you might have a dinner, but you're not making ham and a turkey and a chicken and, corn souffle and green beans almond. You might make 3 recipes out of 100, all right, or out of 50. So in a given cell, you might have 5,000 or 6,000 of those 23,000 genes turned on, and that's what makes that cell function that way, okay? So during transcription, we have to decide, your cell has to decide what's on, what genes are on and turning into proteins, and which genes aren't. Activators are proteins that turn on a gene, okay? Transcription factors in general are proteins that turn on and off transcription, all right? And if you turn on transcription, you're going to turn on the whole process of protein synthesis and make that protein. And if you turn it off, you're going to turn that off and you're not going to make that protein, all right? Or maybe you make less of it. Okay, so activators are proteins that tell RNA polymerase to come land on the gene and make that RNA, okay? Activators attach to or bind to a promoter region. It's right before the gene, and it's the switch for the gene, okay? So activators help RNA polymerase, like maybe you could make the analogy might be like runway lights. All right, RNA polymerase says, "Oh, I can land there because there's runway lights telling it to land there, and it'll make the protein." Okay, it'll make the mRNA, which will then make the protein. Repressors do the opposite. Repressors block RNA polymerase from doing their job. Okay, so let me give you a really quick example that applies to many people right now. All right. Obviously, there's many different skin tones. So some people's skin tones don't change. Particularly if you have very dark skin tone, it doesn't vary much. But let's just take an example of a lighter skin toned person like myself. In the winter, I'm extremely pale. And then in the summer, I'm slightly tanner, but not particularly tan. So right now, I'll go outside, right? I've been going outside and doing yard work for hours at a time. And as UV light hits my skin, repressors come off of certain genes, which were blocking me from making melanin, because it was the winter and I didn't need melanin. And melanin helps to prevent you from getting skin damage. And activators are taking their place. So on the gene for melanin, which makes you tan, right? It makes your skin cells produce dark pigment. The repressors will come off as you get exposed to sunlight, and activators will bind to the gene, the promoter section of the melanin gene, and that will tell RNA polymerase, land on that gene in your skin cells and start making melanin. And guess what happens when this happens? Out in the sun, you'll start to get tanner, okay? And, you know, it takes a while, but you're actually changing what genes are on. So my point is here, you're continuously producing different amounts of protein based upon what your body needs, based upon your diet, based upon your lifestyle, based upon the time of day maybe, based upon the season. Your body turns on different genes based upon big developmental changes, right? How many of you right now can remember a time where maybe you were blonde as a kid and now you have dark hair? Well, the genes for melanin in your hair follicles turned on, you know, who knows, when you're 7 or something, and then you start to have darker hair. Okay, I'll do this and then we'll move on. We actually, when we make this mRNA, before we make this mRNA during transcription, we make pre-mRNA, okay? So just like you would make like a first draft of a paper, and then maybe you chop some stuff out of it because it doesn't sound good, or some run-on sentences get kind of, you know, fixed up. You make the first version of mRNA is really called pre-mRNA. And you remove some sections of it. You splice out some sections called introns, which are not going to be in the final product, and you put together the exons. Exons are expressed, introns are not. Okay, and then the mRNA is the final version. It can leave the nucleus, where it's going to be translated in the next step. Okay, real quick, we'll try this. Here's a gene. If you were doing DNA replication, this is what would happen. You'd read a T and add an A, right? The DNA polymerase would read an A and add a T, okay? If you're doing mRNA, however, that's not how it's going to work. A T up here pairs with A pairs with U, and a C pairs with G, all right? CCC will make an mRNA molecule of GGG. ATT will make an mRNA molecule of U, A, and A, okay? So try this one. If this is your gene sequence, what is your mRNA? Pause it right now. Give it a try. And your answer in 3, 2, 1. Okay, that's it, because I'm almost at 10 minutes. So I'll make another video soon.